session of pulse and basic nephrology by Gopalakrishnan, sir. Sir, can you share your screen? So can you share your screen or do I share the screen?
So Vinith, I will request you to share.
Yes, I'll do that.
No, I would request you to share.
I'm doing that, sir.
Yeah, please share, because my signal strength is not that great.
Yes, sir. Sure, sir. Sure, sir. Is it visible to all of you?
Will you please share?
I have shared, sir. Is it visible?
Fine. Yeah, sure. Yeah, it is.
Yes, sir. We are ready, sir.
Shall I start? Yeah.
Yes, sir. Please start, sir. Please start.
Very good evening. Thanks ISN, and thanks Dr. Shyam Bansal and Dr. Arvind Khanji and Dr. Vinith Behra and Jasmine, and of course the presenter today. And all the attendees, it's always a pleasure to explore and to get excited at the clinical nephrology. Particularly so, the fundamental, the basics, the basic physiology and the perturbations that do happen in the physiology, and understanding a disease, how a disease is actually happening, what happens over there. So this is always exciting. Focus more on these things. These things are not just purely of academic interest. It is not so. Am I audible? Vinith, am I audible?
Yes, sir. You're audible, sir. You're audible, sir.
Yeah. So I have got a very strong conviction that without understanding the basic pathophysiology, future newer therapeutic advances will not happen. That is number one. Number two, for almost every disease, now we have kind of a guidelines and protocols which get amended every quickly adopt to these guidelines, and if you just apply them, and you will be a very successful specialist. But over a period of time, the kind of fatigue will set in. To overcome that, we should always cultivate a habit of looking at science and getting excited about it. So how does it happen? Only if you explore the unfathomable pathophysiology of the diseases only, you will get that excitement. And today also, most of you will be quite aware of this. I'm not going to discuss anything new about it, but still, this is something very fantastic. I used to look upon this particular mode of cell death with a lot of excitement, right? We are going to discuss on NETosis. You all know what is NETosis. It is the drug. Apart from excitement and academics and all that, it is very frequently asked university exam question, be it DNB, DRNB, be it DM. Most of the universities prefer this question nowadays. Okay, what is it all about? Next slide. Yeah. As the name explains, it is a cell death by neutrophil extracellular traps. When I was in MBBS in early 1980s, we just knew only one mode of cell death, that is necrosis. Only in early 90s or late 80s, the concept of apoptosis came in. Even apoptosis, when the concept was introduced, it was so the medical fraternity was, the scientific community was really taken aback. And apoptosis has been happening, even the coronavirus, how does it disappear? How it gives way for the metanephros and so on. by apoptosis only. So day in and day out, apoptosis has been happening in our own system, but we were not knowing it, right? Be it thymic convolution or the atrophy of the prostate and the atrophy of the uterus and so on. All these are apoptosis. So apoptosis is a programmed cell death. Okay. Now, is it only necrosis and apoptosis? What are the other modes of cell death? There are so many. Pyrotosis, ferrotosis, and a combination of necrosis and apoptosis called as necrotosis, right? So like that. So netosis is one type of cell death. So why is it happening? Why is it happening? So it is happening primarily in the neutrophils, but now we know netosis can happen in other cell types as well, particularly monocytes.
Sir, you're not audible, sir.
You are mute.
Sir, you are not audible.
Please unmute.
Gopalakrishnan, sir, you are actually muted. So please unmute yourself. Yeah, now you are unmuted, sir.
Okay. My signal strength is very fluctuating. I wish and pray that it should go uninterrupted. Okay, fine. Am I audible now?
You're audible
Am
now,
I
sir.
audible now? Yeah, fine.
Yes, sir, you're audible, sir.
Yes. So the previous slide, please. The previous slide.
Yes, sir.
Yes, fine. So this is it's another form of programmed cell death. Primarily, it happens in neutrophils. Of course, now we know that the same netosis can happen in other forms of cells as well. So why is it happening, first of all? Why is it happening? Look at the last point, third point I have mentioned. It is evolutionarily very highly conserved process, meaning it has been there for millions and billions of years. Why do we say that? It is not peculiar or specific for humans, not only for human neutrophils. It is there in the animals in the much lower state of evolution. Not just that, even a single cell eukaryote, it has been observed, and in certain plant forms also a different kind of netosis is there. So it is evolutionally very highly preserved, meaning it should have definitely given a survival advantage. Okay, what is the survival advantage? This is the mechanism which protects the host against the invasion of pathogens. Primarily, it evolved for that purpose. Okay, so this single slide, even if you forget the whole lot of it, it is the extracellular. Now something has come out of the cell. It is trying to trap the microbes which is there in the vicinity. So what is this trap? What has come out of the cell, the cytosol? As you can see here, a component of the DNA itself, the chromatin, altered chromatin, studded with so many materials, histones and so many, the cytoplasmic granules and so many substances and peptidases and a lot of things which are there in the cytosol itself and cytoplasmic granules, and most importantly, histone and the DNA. It's all coming out of the cell, trying to trap and kill the organism. So remember, neutrophils, even without this, they can handle. They are the primary line of defense in our immune system. Even without this extra effort, it can just engulf. It can just handle the bugs. But in what situations it does it? It happens particularly in response to certain danger signals, particularly when the situation is a little more alarming. There is a kind of a sensor mechanism in the neutrophil repertoire, and it resorts to this, right? So it is a vital part of the innate immune system. Next slide, please. Yeah. So what are the activating stimuli? There's a big list of activating stimuli, namely so many bacteria, viruses, and fungi, and protozoa, and so on. This is one group. Then It need not be a full organism, a complete organism, even a bacterial cell wall, like lipopolysaccharide of the gram-negative bacteria, that by itself can stimulate NETosis. Second thing. Third thing, a host of antibodies and immune complexes, several cytokines, particularly interleukin-8 and TNF, and several chemokines also can work. Most importantly, certain crystals, very importantly, monosodium urate, a uric acid crystal can stimulate this. So this point you have to remember. The second component of this, what are the stimuli? The microbes, the bacterial cell wall, particularly LPS, and the conglomerate of antibodies, immune complexes, cytokines, and chemokines. And fourth important thing is microcrystals. Next slide, please. Fine. So there are two types of NETosis. Please concentrate the box below first. It's a classical NETosis. It's called suicidal NETosis. What happens when the major portion, a significant portion of the chromatin and DNA comes out of the cell and there is a breach of the plasma membrane of the cell membrane? What will happen? The cell cannot survive. So it is suicidal. It is a classical NETosis. There is one form of NETosis where the trap comes out, the extracellular trap is there, but still the vitality, the viability of the cell is maintained. You can simply forget about this, but why? Because the international nomenclature of the scientists is that any process which does not involve cell death, you can't use the word osis, no? Necrosis, apoptosis, pyrotosis, ferrotosis, and all that. So the vital NETosis and the clinical relevance, we don't understand this. Definitely, it is going to be a future area for research. Why vital NETosis? What is its physiological role? We don't know. So just forget about it. The classical NETosis only we are talking about, and it is suicidal NETosis. Then, next important point you have to remember is that there are two important mechanisms. The most important mechanism is reactive oxygen species, ROS pathway. Without ROS, reactive oxygen species, the NETosis cannot be initiated and sustained. This is the most important, right? The other one is the PAD4 pathway. Please remember this PAD4. Even if it is a bit difficult to remember, please try to remember that this is very important because peptidyl arginine deaminase 4. So PAD4 RAS. Next slide, please. Next slide. Yeah. So how is it happening? The slide may look a bit very complicated, but I'll try to make it very simple, very simple. Look at the center, the top. Due to many reasons, there can be entry of calcium into the cell, or in certain stressful situations, escape of calcium from the endoplasmic reticulum. This is the very first important step. Due to the calcium pores and calcium channels, opening up of the calcium pores, calcium can enter into the cell, neutrophil, I'm talking about neutrophil, or calcium can get extruded from endoplasmic reticulum. So this is the first step. Increase in the calcium concentration within the cytosol of the neutrophil. That is number one. What does it do? The calcium directly can stimulate the mitochondria to produce mitochondrial ROF. I told about reactive oxygen species. Okay, that can happen. Or this mitochondrial ROS can cause activation of the protein kinase C, PKC. That is important step. Please remember that. Ultimately, the most important thing is that activation of the NADPH oxidase. Why this is important? This NADPH oxidase, that is an important source for ROS. Okay, now you understand there are two sources for reactive oxygen species. I said reactive oxygen species is very important for NETosis to happen. There are two sources of ROS. One, the mitochondrial ROS. The other one, ROS that is formed by NADPH. As you can see here, this NADPH induced ROS can again activate the mitochondria to produce further ROS and so on. Okay. Here, if you are really have a lean to understanding the neutrophil, I always used to love to learn more and more about neutrophil. Actually, neutrophil is one cell, one type of white cell, which does not really depend on mitochondria normally, even for its energy source. The primary energy source for neutrophil is not your oxidative phosphorylation, not your Krebs cycle primarily. It depends on glycolysis. In fact, the mitochondrial concentration is little lesser in neutrophil. So, but it is surprising here in the very important process of NETosis, the mitochondria of the neutrophil makes its presence felt and it jumps into the process of NETosis by generating the ROS. Are you clear about it? Next slide, please. So this reactive oxygen species process initiates the process of the trap, extracellular trap. Here, this picture, I will talk about the role of PAD4. Okay. So this is the pathophysiology of ANCA disease. So double purpose actually. When actually this I have to talk about the clinical application. The two important kidney diseases where NETosis is supposed to be playing an important pathophysiological role is one is ANCA disease, the other one is SLE. So this is ANCA disease. Okay. So what happens in ANCA disease, anti-neutrophil cytoplasmic antibody disease, there is a priming of the neutrophils. That's what happens first. So priming with certain substances like complement 5A or lipopolysaccharide, TNF-alpha, IL-8, and so on. So only to the primed neutrophils, the ANCA goes and binds. Okay. But in ANCA disease, always it's a problem. Like you have anti-neutrophilic antibody, but the antigen is very much deep into the cytosol. It is not available on the surface, right? Okay. So only this priming, the process of priming exteriorizes the PR3 or MPO onto the surface. Once it comes to surface, the ANCA binds to that. Next, what happens? There is a reactive oxygen species burst, ROS burst. Right now, we saw just that there are two sources of ROS burst, right? NADPH oxidase and then mitochondria. Once this ROS burst happens, even without PAD4 expression, it can form the trap. That's what the previous slide described. There was no mention of PAD4 there. It is purely ROS pathway. But here, there is a PAD4. I just mentioned what is PAD4, peptidyl arginine deaminase 4. What is this all about? Why should it happen? This PAD4 expression is very important because that causes a very important step in the process of NETosis. What is the process? There is histone within the nucleus, right? The histone gets citrulinated. It adds citrulline. That is done by the PAD4. The citrullination of histone is an important step. After that only, the shape of the nucleus gets changed. See, the multilobate, lobular, beautiful nucleus, it becomes kind of a small or rounded or ovoid shape. The shape itself becomes totally different because this of the citrullination of the histone that alters the structure and the orientation of everything about nucleus. There are now mechanisms in even electron micrograph. You can see the neutrophils. You can differentiate the neutrophil that is undergoing the process of NETosis, looking at the nucleus itself. You look at the first slide, the multilobar lobulated appearance, and here what is happening here. Okay. Then next, what happens is that after this process, when there is alteration of the histone, that leads on to decondensation of the chromatin. Normally, nucleus and chromatin, such a big thread, running into huge lengths, it is nicely packed in a condensed form. Here, after this citrullination of the histone, modification of the histone, that leads on to decondensation of the chromatin. And finally, a very important thing, look at the point number 5. Neutrophil elastase, that gets activated. That breaches the barrier of nuclear membrane as well as the outer cell membrane. The role of Ne is important. Remember this point, neutrophil elastase, that causes breach and the whole of it, lot of it comes out. What does it contain? As you can see here, the chromatin thread and altered histone and so many components of the granules of the cytoplasm and several components of the cytosol. It may achieve the engulfing and trapping of the organism, but at the same time, what happens? All these substances which come out of the cells, they induce local inflammation. They induce destruction of the local tissue. So it is this kind of an avaricious anger on the invader sometimes inflicts a self-injury. That's what is happening here. So your action always has to be kind of tempered. It has to be balanced. In life also, that is the philosophy of life, successful life, right? If that is not there, the harm happens unintentionally. So all these mediators can cause local destruction, local injury, and so on. More importantly, the material, so many components which have been kept deep inside the cell nucleus, they come out, they get exposed. So that induces autoimmunity. Here comes the importance of the role of NETosis in SLE. Next slide, please. So what I said, again, the same thing, no new point here. As you can see here, what are the first, the top, the initiators, the stimulators, the microorganisms, the damaged cell products, LPS, antibodies, urate crystal, I said. All these, they can induce the process, okay? And then the calcium entering from the endoplasmic reticulum into the cytosol, activation of protein kinase C and NADPH oxidase, ROS, and then the histone cleavage, citrullination of histone, and the chromatin decondensation and coming out of it. And here, in every step, as you can see here in the blue small boxes, there are so many names of agents are given. So a lot of research is on in diseases which is mediated by excessive NETosis to utilize the knowledge of the stepwise process of it by addressing that by therapeutic agents. Now, a lot of research is happening on the last one here, the DNase. What is this DNase? It can act on the extracellular trap. It can cause the dissolution of the NETosis. Actually, normally day in and day out, it's NETosis is happening in some people in a small degree, but we escape from disease because there is a beautiful mechanism of removing or disposal of NETosis traps as well. Okay, in these disease states, actually, that the removal mechanism is also hampered. So an important drug that is being developed now is the DNase. Okay, next slide, please. Next slide. Yeah. So it is not just SLE and ANCA disease and so on. As you can see here, these the components of the trap, it can cause activation of the tissue factor that can result in coagulation and thrombosis. This was particularly considered as an important mechanism of the recent COVID pandemic. No, it was a pro-thrombotic state. NETosis was one of the pathophysiological mechanisms implicated in the thrombophilia of COVID, right? And stimulation of cytokines resulting in T cell activation and B cell activation, autoantibody production. That is another mechanism of systemic autoimmune diseases. And more importantly, many components, particularly LL37 from the neutrophil, that stimulates the dendritic cell. You all know dendritic cells activation can result in the formation of interferon. And type 1 interferon is very much implicated in SLE. What is the drug that is tried to address the interferon in SLE? Anifrolumab, right? Anifrolumab. There is a lot of papers are coming up, right? Okay, so interferon. So you name any mechanism of SLE, ultimately by different mechanisms. One by dendritic cell, interferon, SLE, activation of B cells, autoantibody, SLE, exposure of the nuclear components outside the cell and resulting in inducing antibody, and all that happens. And injury to the endothelial cells can result in vasculopathy, a host of diseases, including ANCA disease, and more importantly, antiphospholipid antibody syndrome. Also there it is implicated. Next slide. And one more thing, previous slide, please. Yeah. So look at the orange color, yellow color box on the right side. Please remember this particular point also. When you talk about NETosis and SLE, this particular point you have to remember low density granulocytes. What does that mean? The granular composition and the quantum of the granules within the cytosol will be less. These cells are not usual. Normally, they are very small in number, but there will be significant presence of LDGs. What are these LDGs? They are neutrophils only, but they have got very special characteristics, and these cells have got a high propensity to go for net formation. Next slide. Next slide. Yeah, the low density granulocytes appear in patients with SLE. They've got very unique characteristics, namely, they tend to produce more quantum of inflammatory cytokines, including interferon. And they have got the capacity for production of excess quantum of mitochondrial reactive oxygen species. And the traps which are coming out of LDGs as compared to the the regular neutrophils, they contain more autoantigens and more oxidized mitochondrial DNA. So the traps coming from these LDGs are particularly stronger immunostimulants, ultimately resulting in SLE. Next slide. Thank you so much for patient hearing. Hope it was useful. Yeah.
Thank you, sir. We hope that agents targeting NETosis clearance or promoting